video, I was going to go over the genetic engineering way to grow lab. So in this lab with all signs takeout, you need to read the safety instructions here. And a lot of it is pretty obvious about not eating things, putting things in your eye, making sure to washing your hands. But anyways, this is also a map of everything that should be included in your kit. So in this lab, you will get a case study like you do in all labs with this company. And in this one, you have a 6-year-old child named Jose. He's 35 inches tall. And he's going in because his parents are worried about his height and that he isn't tall enough. So a little bit further down below, there is a line with the average height of children at various ages. And in the 5%, it asks you here to find his age and his height and to graph it and put a dot. On the next one, it asks you what you would conclude about his height. And again, any of this data that you need to know was in this very short reading here that you can find. On page 2, you end up having Jose's doctor suggest a blood test so that they can test various things like the thyroid hormone, insulin, growth hormone, and cortisol. So this is where you are going to pull out your plasma test kit that they included. So here it says use the materials and the instructions. So the instructions are not here on the lab kit. The instructions are actually in the paper that came, it looks like this. It's in the paper that came with the plasma and with the test strip. So what you're going to do is you're going to read through this, and it's going to tell you how to dip it into the vial and how you'll get a reading. Now, I drew up a couple of fake readings here. So these are fake. But then once you get the reading, and I didn't want to give you the right one, so this is a fake one. But once you get this fake reading, you're going to hold it up here, and you're basically going to match it and say, oh, for thyroid hormone, it was red, so that means he's low. For insulin, that was peach, so that meant normal. For growth hormone, it was green, so that meant it was low. And for cortisol, that was yellow, which meant that was normal, if that was your test strip. Here's another example test strip that it could look something like that. And you just find out his level at each spot. You are going to put all of that to the side. And you are now mostly done with that, but you're going to log that here. And then you're going to go down on this problem and explain the results. Then here, so it has a full reading here. This is a picture of the pituitary gland. This is in the brain. It's at the bottom of the brain stem, and it makes the human growth hormones. It talks to you, gives you a little bit of history about how the human growth hormone used to be taken out of cadavers. That means dead bodies. It was extracted from dead bodies. That was gross and limited and potentially infected at times. So they talk about how this is now built or created using bacteria. So that whole reading is here, and you'll need to read that to be able to answer the questions below. So once we get to there, we are on to being a scientist now, okay? So now, well, that other part was being able to read labs. So that was more like the doctor and labs part. But this one is more a lab scientist that's working on genetic engineering, okay? So it talks about growth hormones. It explains chromosomal DNA and then the plasmid. So chromosomes have DNA that's in a larger DNA-looking ring, and it also has smaller ones, although it's not drawn smaller here, that are plasma, okay? It says smaller, but they're not drawn smaller. They should be. So it's going to ask you some questions. Again, all of these answers to questions are right here if you do the reading, okay? And I do expect thorough and complete answers and not just, I'm trying to hurry through this and sloppy throw down an answer. So then this is where you're going to move on to your test strips that are like this. And it asks you to cut them out along the line, which might seem like an unnecessary step, but it becomes more necessary as you go along. But at any rate, you're going to get where you are going to tape up the plasmid here for the bacteria, and you're going to tape it into a loop. An important piece to note on this is that this part of the blue has the AMP gene, which means it's resistant to ampicillin, meaning it can't be killed by ampicillin. You'll understand better why that's important as you go through it. But as you go through, you're also going to cut out your human DNA. Now, human DNA is not in a circle. It's more in a straight line. So with this one, you're going to leave it in a straight line. And in this one, the red area is for human growth hormone gene. The directions are really specific on what you're going to do. But basically, you're going to cut a piece of what's called a restriction enzyme, which basically is something that cuts, it shows the scissors cutting, it cuts the DNA exactly where you want it cut. That's what a restriction enzyme does, is it cuts the DNA where you want it cut. So here, using your clear piece, you're going to find the exact sequence that you need wherever it matches up perfectly with the same letters on your human and on your bacteria. The directions will be very specific in that you are going to use the restriction enzyme to cut part of the bacterial DNA, and you're going to insert it onto the human with the DNA, and you're going to combine that. So the directions are pretty clear. So just make sure that you follow this, that you use the restriction enzyme. Make sure you cut along the dots and you draw it when it tells you to. Use the scissors to cut it using the restriction enzymes and things like that. So follow that. Then it's going to ask you questions like, what do restriction enzymes do? Well, the answer was on the previous page. It's going to ask you what's meant by the sticky ends of this. And again, the reading is directly above it, explaining all of that. Just make sure that you're reading along the way, and you should be fine. So you are going to tape this all together and make a piece of combined bacterial and human DNA to show how these hormones are replicated. So to do that, you're going to, this next step is a little bit different. It says, congratulations, you've successfully engineered a recombinant DNA plasmid. The DNA plasmid is a ring of DNA that combines genetic material from one organism with another. So this is where you are going to take, start this next step, and you're going to need to put the recombinant DNA, which is the two DNAs that are together. Now, I didn't do this part, but these two will be combined, and I didn't want to show you how that was doing it. It's pretty clear how to do that, but you're going to stick it inside of one of these envelopes, okay? So then you're going to have bacteria that don't have it and bacteria that do have it. Then you're going to simulate that you released the ampicillin, which killed the bacteria that didn't have this gene, which means the one that's empty is going to be killed. So you'll toss it to the side and you'll keep using this one here. So it explains that whole process and how that works, called a transformation. It explains that on page 9. So you're going to read through the steps. But essentially, there is bacteria that's resistant to antibiotics, and they use a calcium chloride treatment to open that bacteria up and allow other things to come in. So then they will add the plasmids in with the correct DNA, and then that will be a new transformed bacteria. So this is a newly recombined or built bacteria. And then they'll grow the bacteria here. You need to summarize that all in your own words and write it out down at the bottom here. So we will move on. Here there is just some more information about what I already explained about putting the bags to the side. Some more questions at the bottom. And then it talks about how this is done with human growth hormone and how this happens with RNA and how these types of therapies are used to treat people in different situations. Okay. The last part is that now you have this human growth hormone, and you are going to take this human growth hormone and just set it on the bottles as part of like the simulation of now these bottles have the correct human growth hormone in it. You're going to come to the end. You are going to put the steps in order of the genetic engineering process. So number 1 and 8 are done for you. Just because I know you were really concerned about Jose and his height, at the end, they're going to tell you about how the hormone therapy helped or did not help him or how it affected him. Just as a quick summary on Jose, there's nothing you have to do there, but I know it'll make you all feel better about Jose's situation. And then you will go to making your own insulin. So this section is mostly a reading section, but a reading and then writing. What they want you to do is they're giving you the information here about how this recombinant process, recombining or genetic engineering DNA process works. Then they're going to ask you to write an essay here and use these words and underline them. I would really appreciate it if you did underline or highlight that you did all, like incorporated all of these words. But the picture really keeps you on track with what's happening. So the human DNA is pulled out, just the necessary pieces are cut by the restriction enzymes to get the exact insulin pieces that are needed. Those are incorporated into plasmid DNA that's pulled out of the bacteria. Then they're recombined. They're put back into the bacteria, and then the bacteria is replicated. There's a lot of little nuances that I left out of that explanation that will use all of these words in the reading. So I just gave you a very basic, non-sophisticated way of explaining that. So you need to add in all of these words. That's it, and that's how you complete the Way to Grow Genetic Engineering Lab.